So the problem we were trying to address in this work is really just making flu vaccines better. So, we have annual flu vaccines because the virus mutates every year. And so the antibodies that you made in response to last year's vaccine may or may not protect you against next year's virus. So what we and many others in the field are trying to do is take steps towards what's called a universal flu vaccine that could provide protection against a very broad variety of influenza viruses, including next year's. The two major antigens, surface antigens of influenza viruses are hemagglutinin and neuraminidase. Hemagglutinin is kind of like the coronavirus spike. It's the protein that binds to a receptor on your cells and then facilitates entry of the virus by merging the viral membrane with the host cell membrane. Hemagglutinin is also the major antigen in commercial traditional influenza vaccines, and antibodies against the hemagglutinin are what provide protection. Our broadly protective influenza vaccines are based on a computationally designed nanoparticle platform that we've developed here at the Institute for Protein Design. What these nanoparticles do is they repetitively array the antigen, the protein from the virus that you're trying to mount an immune response to. Repetitively arraying that antigen tells the immune system that this is something dangerous. So repetition is actually viewed by the immune system as a danger signal. Viruses themselves are symmetric, repetitive arrangements of proteins. Bacterial cell walls have repetitive chemical elements. So your immune system has evolved to respond strongly to repetition. So what we did was we took our computationally designed nanoparticles. It's kind of like playing Legos with proteins. We programmed them to assemble to very specific structures. And we attached the hemagglutinin on the surface of those nanoparticles to mount a robust response. The next trick we played, however, was we took advantage of the unique capability of our nanoparticle platform to co-display multiple different hemagglutinins on the same particle. And what we found is that either nanoparticle vaccine format gave responses against the current seasonal influenza strains that were equivalent or perhaps just a touch superior to current commercial vaccines. That's very important from a regulatory perspective. You need to be at least as good as the standard of care. But the special thing, what we saw in addition, was that we got much broader responses, and these broader responses were protective. So the vaccines actually protected against H5N1 bird flu or H7N9 influenza viruses, whereas current commercial vaccines did not. So we're thinking these vaccines may be what you might call a supra-seasonal vaccine, a vaccine that protects for more than 1 year, right, or provides high efficacy protection across multiple years. The key takeaway is that this virus provides really good protection against seasonal strains and potentially broader protection against next year's strain or even 3-4 years from now. So that's the one key takeaway. What we're doing next is we're actually evaluating this vaccine in a phase 1 clinical trial that's scheduled to begin next month at the NIH. So this entire project has been in close collaboration with researchers at the NIH's Vaccine Research Center, specifically the labs of Barney Graham and Masaru Kanakiyo.